Scypha, also known as cycon, exhibits a complex morphology reflecting its cycanoid canal system, an advancement over the simpler asconoid type found in some other sponges. 1. Overall shape and size. Scypha typically possesses a cylindrical or vase-like shape, often described as resembling a pineapple. Its size ranges from 2.5 to 7.5 centimeters in length, though some sources cite smaller sizes, around 2 to 3 cm. The sponge is usually attached to a substrate such as rocks, shells, or corals in shallow marine environments. 2. Body wall structure. The body wall is diploblastic, meaning it consists of two main layers, an outer penicoderm dermal layer and an inner choanoderm gastral layer. Between these layers lies a gelatinous mesohyl mesenchyme containing various amoeboid cells. The penicoderm comprises penicocytes, flat cells forming a protective epithelium, and porocytes, specialized cells creating the ostia, incurrent pores, allowing water entry. The choanoderm lines the radial canals and is composed of choanocytes, collar cells, each bearing a flagellum that generates water currents for filter feeding and respiration. The mesohyl houses amoeboid cells involved in skeletal support, nutrient storage, and reproduction. 3. Canal system, cycanoid. The cycanoid canal system is a key feature distinguishing Scypha's morphology. It's more complex than the asconoid system, with water flowing through a series of canals. Incurrent canals led from the ostia to radial canals. These radial canals are lined with choanocytes and are responsible for the majority of the filtering processes. Apoples connect the radial canals to the spongocoel central cavity. Finally, the spongocoel is drained through the osculum excurrent pore at the apex. This system ensures efficient water flow for filter feeding and gas exchange. 4. Skeletal structure. Scypha's skeleton is composed of calcareous spicules, typically needle-like or spear-shaped. These spicules provide structural support and project from the body surface. Different types of spicules can be present, including monoxin 1-rayed, triaxin 3-rayed, and tetraxin 4-rayed spicules, with specific arrangements depending on location within the sponge. 5. Osculum. A prominent osculum excurrent pore is located at the apex of the sponge, serving as the outlet for the filtered water. It's often surrounded by an oscular fringe, a collar of spicules that may prevent the entry of larger organisms. In conclusion, Scypha's morphology is characterized by its cylindrical shape, diploblastic body wall with a cycanoid canal system, calcareous spicule skeleton, and a prominent osculum. This intricate structure reflects its efficient filter feeding and respiratory strategies in its shallow marine environment.